Welcome to this class. In this class, we will look into why rodent models, why rodent experiments are required, what kind of experiments we have been doing, and how we can design several kind of devices. What are the existing commercial devices? We will look into some of the microelectrode arrays that are available in literature. And then we will see that what kind of techniques that are used for fabricating these devices. Okay. So, the first question is why directly we do not go for humans, right. So, why rodent models, why we do not go even for non-human primates? Because of the ethics. We need to prove that the technology or the device is performing well before we go for larger animals. And also generally rats are used as a model for mammalian brain, although it is not as developed as monkey's brain. So, for certain application we can use rat's brain or rodents. For some application where we are looking at the fine motor movements, right, like see this is motor, right, because whatever we are walking is a motor movement, this is a motor movement, this is a gross motor movements. When we hold the pen is a fine motor movement, because we use our fingers to hold the pen very delicately. I will hold the pen and take it up, and then I can move it, right, I can maneuver it whatever I want to do. So, now this is a fine motor movements. For patients who have or whose brain is injured in an accident or whose spinal cord is injured, generally the motor area is affected, and the recovery, in the recovery phase, the gross motor movement, the bigger motors, right, these moments is possible to recover like this, right, even walking is possible to an extent. But the fine motor movements are very difficult, and that is why you see all these peripherals, prosthetics, right, that are used so that it can help the patient to do some daily routines. But why we cannot recover the fine motor movements, right? What is the anatomy, how the signal flows, right? These all things are very important for us to understand. So, if you want to look into those kind of research where we are looking at the fine motor movements, then non-human primates are better, because monkeys we can see the motors movement, right. Rats paws is kind of difficult. So, quickly we can do some experiments with the rat and then move on to non-human primates. But if you want to understand some effect of drugs, efficacy of drugs, anti-epileptic drugs for example, right, then we do not have to really go for humans, right, because it is not possible directly go to humans. We can start with the rodent models. So, if you see the slide, why rodent experiments is because rats, one, are used as a model for mammalian brain. Second, some of their brain structure resembles the more primitive elements of human brains, and the research suggests that rat more accurately reflects human physiology than other species, mimicking human disease more accurately in many cases. Okay. So, the flow for implantable device research starts from the rat. Now, more or less a cat is not allowed for research, but guinea pigs are allowed, rabbits are allowed. We go for non-human primates, and finally, we go for the human studies. There is a flow for implantable device research. Okay. Anatomically, again Shabri will discuss in detail, but if you just understand very top view, then we have, let us see this B1, okay, frontal lobe, all this one is your frontal lobe, then we have parietal lobe, this red one PL, parietal lobe, then green one temporal lobe, and finally this one is a occipital lobe, alright. Frontal lobe, parietal lobe, temporal lobe and occipital lobe. This we are talking about human brain. Further you want to understand, then there is a cerebellum, thalamus, hypothalamus. This is your forebrain. Okay. Three things: one, cerebrum or cerebrum, second is thalamus, and third is hypothalamus. Then we have corpus, midbrain, and then the brain stem, it is cerebellum, pons and medulla. So, this is the internal view of the right hemisphere of human brain with three main parts: forebrain, midbrain and brain stem. If you see this particular schematic, which is in C, you see how the human brain and rat brain is compared. Different dedicated functions, for example, initiation of motor actions, which is primary motor cortex, which is the blue color, okay. And same thing goes for the rat's brain also, it also has a motor cortex. Then we have somatosensory cortex or perception, which is for primary somatosensory cortex, which is red color in human brain. In rat also we have the same thing. And then we also have the visual cortex, right, which is the occipital region here, same way here, and then we have the olfactory cortex, you see here, you see here, right. So, primary motor cortex, primary auditory cortex, the yellow one is auditory cortex, here also is there auditory cortex, right. Then we have primary somatosensory cortex, then we have primary visual cortex, and finally olfactory cortex or bulb, olfactory bulb. Okay. So, these are the anatomical representation of human and rat brain. Okay. Now, let us go to the next slide. So, why we are talking about all this region is because we will show it to you where we implant the device in which region. Okay. That is the reason of we showing it to you, the anatomy of the human brain and comparing with the rat's brain. So, we will look into the following experiments at a minimum, or the technologies that we have developed at a minimum, and understand how can we fabricate neural implants, right, implants that can be used to acquire signal from the brain. Whose brain? Rat's brain, right. So, for that these are the recorded signals, ECOG signals. This is a OpenBCI tool, right. This is a Cytone Daisy biosensing board. Then we have earbars, EIB holder, rat adapter. Actually the schematic shows a implant or a device that is implanted onto the rat's brain, and we able to record the ECOG signals from its brain. Okay. So, as I told you, ECOG is nothing but electrocardiography, and we can study the effect of anti-epileptic drugs, AEDs, anti-epileptic drugs. Okay. So, we will discuss in detail, but the point is one of the application of this implantable devices is to study the efficacy. Second, I told you about this shank, right. This this shank is a micro electrode array, because one shank consists of 13 electrodes, and this shank is again implanted into the rat's brain, and the data is collected, and we study the efficacy of AED, we can study other things as well. We can also see a PCB that is connected or attached to the MEA. Okay. This red is recovered, okay. Do not worry about this color, actually it is recovered. We use certain chemicals for surgery or during surgery, right. Because of this the color is that like that, but the red is recovered now and the device is implanted and PCB is attached to the device. So, whenever you want you can connect the cable and start collecting the data, right. So, that is the schematic of the device implanted onto the rat's brain. Okay. We will also talk about the Cyton Daisy board and how to collect the signals. I have prepared two classes which will be taken by the TAs talking on the programming aspect of the Cyton Daisy board and how to use it for the application. Okay. Let us go to the next slide. Now, here we will look at the commercially available microneedles, right. So, the question is, if there is a commercially available microneedle which you can see here, single shank with tetrodes, 4 electrodes together are called tetrodes, Tetrodes, tetrodes. Okay. So, multiple tetrodes you can see on the single shank, there are multiple shanks, 3 shank electrodes, you can see this is a linear array of electrodes, is not it. So, all these things are available. What is the purpose, why we need to fabricate devices for our studies? The reason is that each shank, right, each shank, for example this shank, right, it is costing 5,000 US dollar 5,000. Let us keep 80 rupees as $1, we are spending 4 lakhs per shank for one shank. And if some of the electrodes are not working, let us say some of the electrodes is not working, then the company sell this as a class B and that is costing US dollar 700 approximate. Okay. This is not exact price, there will be some changes in the company's policy that I am not aware of. Whatever the last little bit of information we have, we are just putting it here. Okay. But you can always go to these companies, Neuronexus, Thomas Recording, NeuroPixels, right, from this company OpenFSys. So, the point that I am making here is that one is it is costing very high, second is that we need to rely on the imports, right. How to reduce our reliability on the imports, particularly in the medical device technology area, that is what we should focus on. And to reduce the reliability of our country on importing these devices, we should also attempt on manufacturing this kind of devices. And with that is one thing, but another thing is that if you want to have customized needles, customized shanks, customized devices, that cost very high, extremely high, we cannot even buy. When say we is not all of us, some of us may be able to buy, but not majority of us, right. So, can we develop a technology that is affordable in nature, is at par with the technology that is available in the market, and we also become self reliant, right. When we can manufacture these things right here in our country, we should try and attempt that in some years we will be able to be at par with the technologies available commercially, is not it. So, with that goal we started fabricating this device. To your surprise, this $5,000 that we are spending on one shank to import, right, the cost of each needle that comes to us is $50, not 500. and for sure not 5000, right. Is it working? Yes. Is it as good as the commercial one at this point? No. Were we able to acquire all the signals from that region of our interest? Yes. Can we make it better? Yes. Can you reduce the cost further? Absolutely yes, right. So, until and unless you do not attempt, right, how can you reach or how can you achieve your goal? Without attempts there is nothing. When you attempt, there is a failure. When there is a failure, there is a success, right. Everything is interrelated, right. If you do not attempt, there are no failures. If there are no failures, there are no success, right. So, the beauty of attempting to microfabricate devices and sensors is that when you get it, right, when it is successful, you can make thousands of those. For optimizing it, it takes some time, alright. So, but we should try. So, this is one of the commercial device, right, that I have shown it to you on this slide. If you see the slide again, and without demeaning the work or the availability of the needles around the world, this is a very good technology, okay, and it is not so easy to develop as well, but the idea is $5,000 is extremely high for one single shank. So, you see the slide, the another thing that you need to understand is that you see this, there is a electronics and there is a cable that we connected further. This is the figure from IMEC, right, it is copied from IMEC, and okay, you can see here there is a recording module, so the whole electronics is there, and then finally the shank is here, right, and these are electrode arrays which you can see here, alright. So, this can be used for certain region in the brain. If you calculate correctly, this is the hippocampus. So, you can just design such that the electrode goes like this, right, like this, and this is longer one. So, maybe that region is hippocampus, maybe some other region, but the point is that we can also design similar kind of electrodes right here. Okay. What are other flexible microelectrode arrays that are available in literature? So, one of the arrays you can see here, it is for ECOG measurement. The A, which is this figure, shows the exploded view, where you can see there is a silicon and then there is SiO2 on silicon, right. This is highly doped silicon nanowires or nanowells, and on that you have the bioresorbable substrate on which it is designed. You can have degradable substrate, right, a bioresorbable substrate, and you can measure the ECOG and the channels, number of channels, right, are 4 and 256 channels at the bottom, right. So, the point is that people have already attempted such kind of devices, okay, and in fact not just attempted, but also placed on the rat of the brain, you can see here, right, and acquire the signals. You can see that when is asleep, what is activity, right, when we measure with the cortical electrode, what are the activities versus bioresorbable electrode, when there is the when there is a spiking activity, what are the signals and how it looks like when we compare with the control electrode versus bioresorbable electrode. I am sorry, I said cortical, it is control electrode, okay, control electrode versus the bioresorbable. Same thing when there is a seizures, right, like spiking activities, how it looks like, these are spikes, no, it is a seizure. So, how compared to control electrode it looks like, and then can you understand the fast spin, their oscillations, what are the theta bursts, are there any theta bursts, because like I said that we can divide these signals into several frequency bands. So, anyways, the whole idea is to show that the bioresorbable devices can be designed, can be fabricated and can be used for acquiring the neural signals, including activities like seizure activities which are in forms of spikes. But the question is how to make these neural probes, is not it? So, we have something called thin film deposition, in which we have two techniques. First one is called physical vapor deposition, and second one is called chemical vapor deposition. Okay. Physical vapor deposition and chemical vapor deposition. These are the techniques to deposit, for depositing different materials. It can be metal, it can be semiconductor, it can be insulator. If you want to deposit, you can use one of these techniques. So, now in the physical vapor deposition technique, we have three techniques. The first one is called thermal evaporation, second one is called electron beam evaporation, and third one is called sputtering. Thermal evaporation, e-beam evaporation and sputtering, and we will understand each of this technique in detail in the next classes. While in CVD there are several kinds of CVD. There is called low pressure chemical vapor deposition, there can be plasma enhanced chemical vapor deposition, there is a atmospheric pressure chemical vapor deposition, there is a thermal ALD which is atomic layer deposition, and so on and so forth, and these are all micro and nano fabrication techniques. How it works we will see in the following classes. Now, only depositing metal would not be enough. So, we pattern the metal, what do you call? We pattern, P-A-T-T-E-R-N. How can we pattern? We can pattern with the help of lithography. What do you call? Litho, L-I-T-H-O, graphy, G-R-A-P-H-Y. Okay. So, lithography is a word that comes from a Greek origin, lithos and graphics, so as to mean that carving from single stone or carving on stones, right. So, with lithography, if I use UV, I will say it is a photolithography, photolithography. Okay. Photolithography is a technique by which we can pattern or certain metals, materials, and it includes several different sub techniques. For example, in lithography we have to use a mask, we have to use photoresist, and the exposure time, etching, whether it is wet etching or dry etching, right. So, the several things are associated with lithography, so as to finally pattern certain materials of our interest. Then we look into etching, whether it is anisotropic etching, whether it is isotropic etching, and we will also look into machining, where it is called bulk micro machining or it is called surface micro machining, alright. So, all these techniques will go step by step in the next class. We'll start with the silicon wafer, how the substrate looks like, how we can say 100, 111, then we will go one step and go for deposition, then we go one step further, we understand the types of deposition and the principle behind it, the limitations of each deposition, then we go for lithography, and then we understand micro machining, and then we start with the fabrication of different devices. In between we will also talk about the systems that is headband for neonates or headband for adults, or understanding the epilepsy from 1020 system, right, and also understand a bit on what are the EEG signals, ECOG signals, EMG signals, and how we can acquire this and what are the signatures in those signals. So, with that, let us finish this particular class. I will see you in the next class. Till then, you take care.